Okay. Okay, so this is the last thing, right? All right, y'all ready? Happy Friday. What did one toilet say to the other toilet? You're looking very flushed. that is classic fourth grade humor. Classic fourth grade humor. Okay. So we're talking about receptor mediated activity, right? And we're starting with our first messengers. And examples of first messengers are what we ended on last time. Hormones, right? Hormones. So I'm going to give you kind of an example. We're gonna walk through one type of first messenger. So this first messenger is is nitrous oxide. So people have oftentimes tried to alter this pathway in order to affect heart disease, right? So when you're having a heart attack, right, what's the problem? You have clogged blood vessels leading to heart muscle tissue, right? So one way that you can temporarily fix the situation is to relax the blood vessels, right? So if you relax the blood vessels, so you have vasodilation, you have time to go and get a stent put in or whatever it is, right? It doesn't fix the problem, but it allows you to survive and prevents your heart tissue from not getting oxygen for extended periods of time, dying, and then your heart can't function, right? So it's really important. So in this pathway, you have this enzyme called nitrous oxide synthase. And so it will actually deaminate arginine to get to citrulline, and it will produce nitrous oxide. So that will actually activate what's called guanidinosyl cyclase. So what guanidinosyl cyclase will do, it will turn Gtp into cyclic GMP. And a lot of times we have second messengers like cyclic GMP. This is an example of everyday of a second messenger, right? And what's the role of a second messenger? To link things, right? And to amplify the signal. Okay, so that's what we're looking to do. So once we have cyclic GMP will work on a protein called protein kinase G. And so when cyclic GMP is activated, protein kinase G is activated, and it will result in vasodilation, which is what we want, right? For people who have heart disease. So there was a drug that was designed in order to manipulate this system that prevents this cyclic GMP from getting regenerated to GMP and then to Gtp to start all over again, right? So basically when it's in the cyclic GMP form, it's active and you are causing vasodilation. When it's no longer cyclic, when it becomes linearized, then the signaling stops. So what would happen to the blood vessels? They would contract, they would constrict. So it's a little blue pill. Anybody know what the common name of this little blue pill is? It's Viagra. It was actually designed as a heart disease treatment drug, but it's not very effective. It's moderately effective, but it had this weird side effect, right? And why did it have this weird side effect? Because it promoted vasodilation, extended vasodilation, right? So that's how Viagra was discovered. Didn't you always want to know? That's a fun fact. You could share that at your next dinner table conversation. All right, so we talked about first messengers, right? Now we're talking about second messengers. So second messengers are inside the cytosol, and they are there to really, really amplify signals that were generated at the receptor, amplify them, and take it to the rest of the cell, the interior portion of the cell. And so there are quite a few different ones. We talked about cyclic GMP. So anytime you see that little C, what that is saying is that it's a cyclic form, right? So cyclic AMP is the cyclic form of AMP, or you take ATP, you hydrolyze the two phosphates off and turn it into a cyclic form, right? Diacylglycerol is an example of another second messenger that you're probably not familiar with. Inositol triphosphate, IP3, is another second messenger. And calcium is another second messenger. So do you need to know what the second messengers are? Yes, you do. Yes, you do. So we're going to talk about them. We're going to start with cyclic AMP. So I'm going to give you an example. And in this example, this is what happens when you don't eat for a while. Okay, so if you're going to go for a while without eating, what happens to your blood sugar? It drops. When it gets to a certain point, your brain needs glucose. So what do? You have stores of energy in the form of what? Glycogen. You have glycogen storage, right? You have some in your muscles, you have some in your liver. So the body is going to take glycogen. What is glycogen? I'll take an organic, right? So it's glycogen. Insanely long chains of glucose that has branching, lots and lots of branching, right? It is thousands and thousands and thousands of glucose units inside of glycogen. And we'll talk about that later. We'll talk about it in detail. But the point of having glycogen is so that when your blood sugar gets low, we can chop up the glycogen and produce what? Glucose. And we can export the glucose to your bloodstream and we can feed the brain. Yay, your brain's fed, right? Okay, how does that work? How does that work? So we have our receptor that gets activated, right? Our first messenger would be what in this case? Glucagon, right? That's a signal that our blood sugar is low. So we would have glucagon that binds to our receptor, activates the receptor. We have some signaling things that happen near the membrane, which we'll talk about later. It is going to activate an enzyme. So this enzyme is going to have typically kinase activity. And it's going to change, for example, GMP into, well, really GTP or ATP into cyclic AMP, right? So that cyclic AMP is our second messenger. Our second messenger is then going to go and have an effect on downstream signaling targets, right? And so it cascades this way. So if you had one of this original enzyme here that modifies our second messenger of cyclic AMP, right? If you have one of those, it's going to make 100 cyclic AMP molecules. Each one of these 100 cyclic AMP molecules is going to go in, and this is usually PKA, sorry, protein kinase A, right? So it's an enzyme that will phosphorylate certain things. So it's going to go in and it's actually going to make about 50 of them active. Of those 50 PKA molecules, the next enzyme in the pathway is going to end up making 5,000 active molecules. These 5,000 active molecules end up creating 50 million glucose molecules being released, right? So we went from 1 to 50 million. That's a signal amplification, right? And that's just one glycogen, glucagon molecule binding to one receptor. you get 50,000 glucose molecules being released. That's insane. That's insane. But because we have this signal amplification that occurs at our messengers, we can do that. Okay, so you're going to see this over and over again. We'll say this turns into this, it turns into that, but it's not a 1 to 1 to 1 to 1. That's what you kind of have to remember. It's never a 1 to 1. You're always starting with one and then activating more and more and more and more downstream. Okay, so here are some other second messengers. Remember, we said our second messengers were diacylglycerol and inositol triphosphate, right? IP3 and DAG. You have to start getting familiar with the abbreviations because otherwise you spend like 10 minutes just saying the words. So where do these come from? Where do these 2 second messengers, right? These are 2 second messengers. Where do these come from, right? We know where cyclic GMP, cyclic AMP come from, but where do these second messengers come from? Well, remember in lab when you purified phospholipids from the membranes of cardiac tissue, right? You remember one of them was phosphatidylinositol, right? So phosphatidylinositol biphosphate, just has 2 additional phosphate groups, is a membrane protein, a membrane lipid, excuse me, that contains both of these 2 second messengers, but they're hooked together and they're hooked to the membrane. So they're stuck in the membrane until they become activated. So you have to have some upstream receptor activation that leads to activation of this enzyme called phospholipase C. So you have receptor activation that activates phospholipase C, PLC. PLC is going to cleave this whole thing into 2 second messengers of diacylglycerol and inositol triphosphate. So now we have 2 second messengers that are generated from 1 receptor activation. And each of these 2 second messengers has their own role. You ready to see what that is? I know you're on pens and needles, right? All right. So what we just said before is that you have receptor activation. That receptor activation activates PLC. So that's right here. This is PLC. Receptor activation activates PLC, phospholipase C. Once phospholipase C is activated, it takes our phosphatidylinositol and it cleaves it into our 2 second messengers, DAG and IP3, right? So each of these 2 second messengers has its own unique pathway that it follows. Diacylglycerol will then go on and activate protein kinase C, right? And we already saw a protein kinase C before. And protein kinase C is a kinase, so it can go in and phosphorylate downstream targets, right? Anytime you see kinase, you know what it goes and it can do. The IP3, on the other hand, will follow a different pathway. It will actually activate calcium channels. So normally these channels are basically closed, and when IP3 binds to the channels, it opens the channels. Once those channels are open, calcium is released into the cytosol. Into cytosol. Now that you have free calcium, the calcium will complex with what's called calmodulin, and calmodulin and calcium will bind to your target protein, which then induces some downstream effect, right? But at the same time, this calcium can also act to activate PKC. So do you see how much more amplification you're getting? The more second messengers you have, the higher the amplification in the signal. And it's all this pathway of phosphorylation and activation, or dephosphorylation. We'll see some examples of dephosphorylation too, right? So we're going to back up just a second, and we're going to talk about the different kinds of receptors that are going to activate these downstream events, right? So the book separates them, and it gives each class of receptor a separate chapter, well, separate section, and talks about the pathways of them. So that's what we're going to do. So the first one are G protein coupled receptors. That is a receptor that is coupled to, guess what? G proteins, right? That's why they call it that. So they call them Gpcrs. Does it mean that you are PCRing anything? No, unfortunately, they just share the same abbreviation. Okay, so a G protein coupled receptor, they abbreviate Gpcr. So what happens when this receptor becomes activated is that it will disassociate from those proteins that are normally associated with it. So usually you have this receptor and you have all this other junk that's associated cytoplasmic side with the receptor. When the receptor becomes activated, it undergoes a conformational change and all that stuff disassociates. Once it disassociates, it can go and do other things in the cell, right? It can activate other pathways. And part of that, in that scheme of signal transduction, we are going to activate adenylate cyclase, which we said is what? In the pathway, adenylate cyclase will create our second messenger, right? Adenylate cyclase is going to make cyclic AMP, right? What is PLC going to make? It's going to take IP3, it's going to turn it into DAG, and IP3. So these are all what? These three things are all what? Second messengers. Right? Okay, so then you have a second class of receptors. The second class of receptors are receptor tyrosine kinases. So you have a receptor that in and of itself is a kinase. So it is an enzyme, right? It's a kinase, it's an enzyme. Typically in the unbound form, it's not active. But when it binds to its ligand, it undergoes a conformational change, and then it can phosphorylate other things. And because these other things are phosphorylated, now they can participate in binding and induce downstream effects, right? So G protein coupled receptors aren't kinases, right? So when you're looking at these, I want you to kind of think about what the name means and what that's going to mean in terms of your signal transduction cascade. Okay. Tumor necrosis factor receptors, we're not going to spend a whole lot of time on these, but basically they exist on the membrane as three separate components. When they're unbound, in their bound state, they get together. And because of that trimerization, they can induce downstream effects of inflammation and apoptosis. And so it's really, really interesting, but we're just not going to spend a whole lot of time on it. The fourth class are nuclear receptors, right? So we know that these bind to ligands that can go right through the membrane, don't have to be transported, right? And these receptors interact with DNA. So they create protein DNA and protein-protein interactions, right? Because you're going to have this transcription factor that's got to bind to other proteins in order to promote transcription. So you have to be able to bind to the DNA, and you also have to be able to bind to other proteins. So binding of the ligand will regulate either binding of other proteins, of the transcription factor to other proteins, or of the transcription factor to DNA. And then the last one is a ligand-gated ion channels. Ligand-gated ion channels are really cool because they're the primary way that synapses work, right? So if you're talking about a nerve cell, right? At the end of the nerve cell, we call that the, axon terminal. It's the axon terminal. And the axon terminal has these vesicles with neurotransmitters inside of them. And when they receive the action potential, those vesicles will merge with the membrane and release their neurotransmitters to the synaptic cleft, which is the junction from one nerve cell to another nerve cell. And so when those neurotransmitters hit the next cell, they're actually going to bind to ligand-gated ion channels. But these ligand-gated ion channels are, by definition, receptors, right? So when they bind to the ligand that came across from the previous nerve cell, then they're going to open the channel and let one of the ions in. It depends on what kind of synapse it is. Will let the ions in, which will then induce a propagated change, which allow that next nerve cell to fire. So it's really, really interesting, right? So what do some of these look like? These are all membrane bound. Do you see the first three? We have G protein coupled receptors. So here's your receptor in dark blue. It's bound to a red ligand. And normally, right, it associates with what's called a heterotrimeric G protein. So here's a G protein. We have 1, 2, 3 different pieces. So that's why we call it trimeric. And do they all look the same? No, so they are heterotrimeric. Okay, the next one are receptor tyrosine kinases. So typically these exist as dimers, and they'll bind to the ligand, they'll dimerize, and then, now, here's the cool thing about receptor tyrosine kinases. They can either phosphorylate a target protein, or they can phosphorylate each other. So like this receptor can phosphorylate this one, then this receptor can phosphorylate that one. And now that we have phosphorylated ends, we can start to associate with all kinds of downstream targets. And we call those downstream targets adapter proteins. Then you have the TNF alpha, the TNF receptor family, where you have them, these three. So you can see you have three. Normally they're separated, but when they're bound to the ligand, they get together. And then you have these domains within the receptor that once they get together, they induce apoptosis. And so we call them death domain adapter proteins. So when all three are together, they have these adapter proteins that can bind to them and induce apoptosis. So we call them death domain. They got some really interesting names in that section. The fourth one we talked about was our nuclear receptors, right? So they have to be able to interact with DNA, and a lot of times have to be able to have protein-protein interactions. So once you bind that, you can have that. And then the last one was our ligand binding channel. So this is that synaptic cleft. So here's your neuron, and stuck in vesicles, right, are all these neurotransmitters. And when you have this electrical signal that comes through the cell, it tells these little vesicles to merge with the plasma membrane. When the plasma membrane merges, it dumps out a neurotransmitter. In this case, it's acetylcholine. And now this can be between two nerve cells, or in this case, this is an example of a nerve cell activating a muscle, right? So you have the acetylcholine that goes across, and when bound, right, when bound to our channel, now the channel's open. Do you see here where it was unbound? The channel's closed. But when it binds, channel's now open. Now all these potassium and sodium ions can go through, and this is what activates muscle contraction. All right. So now we can start the fun stuff. The fun stuff is where we talk about each of the individual receptors in extensive detail, right? So the first one we're going to talk about are those G protein coupled receptors. So we call them Gpcrs. What you're going to see is that a lot of our sensory perception is controlled by Gpcrs. So there's a conserved conformation of these proteins within the plasma membrane. Typically, they have 7 transmembrane alpha helices. So each of these cylinders that you see represents an alpha helix. And they call them serpentine, right? Because if you start, so here's the N terminus. So do you see how the N terminus is in the extracellular space? N terminus, right? So here's one, you go into an alpha helix, it goes to the cytosol, you have a little loop that you can't see, and then it goes up to the second alpha helix. You have a little loop, starts the third alpha helix. You have a little loop, you go up the fourth, you have a little loop, you go down the fifth. So do you see how it like snakes in and out of the membrane? That's why they call it a serpentine receptor. And so what happens in all of these serpentine receptors is that you have an N terminus that is in the extracellular space, and you have a C terminus that's in the cytosol, right? Okay. And so you can see on these G protein coupled receptors is that they have lots of carbohydrate groups that are attached to the proteins. They call them glycoproteins, right? So this is an example of a glycoprotein because it's a protein that has basically some form of sugar attached to it. And so these help to regulate the receptor's activity. So they function as functional groups. The animation is awesome. Go watch the animation. Go watch the animation. Okay. Before we talk about the whole pathway in detail, there are going to be tons of things that are abbreviated. And you need to start to get familiar with all of these abbreviations. Now, I am telling you that I will give you this table in your exam resources. But if you have to go and look up every abbreviation, it's going to take you too long. But if you're on the exam and you forget, oh my God, what did that stand for? Go look it up. Okay. So it'll be on like the back of your exam resources, right? So we're going to talk today, right, about some of the G protein, the things that are associated with the G protein coupled receptors. We're going to talk about androgenic receptors. So we're going to talk about a bunch of these different portions. So we're going to take one example to start with. One example of a Gpcr is the beta-2 androgenic receptor. So the beta-2 androgenic receptor, if you go back here, this is the beta androgenic receptor, the beta androgenic receptor kinase, right? So when you're looking at these abbreviations, kind of keep it next to you as you're studying. So the ligand for the beta-2 androgenic receptor is epinephrine. When do we produce epinephrine? Fight or flight, we make epinephrine, right? So it's very typical in that when epinephrine binds to the beta-2 androgenic receptor, it's going to create a conformational change. Where in the receptor is that conformational change the most important?
Inside the cell.
Inside the cell, in the cytoplasmic portion of it. So if we look at this structure, what you're going to see is that the dark blue is no ligand present. The green is where the ligand is bound, right? And we call this an inhibitor because we can't actually crystallize it with its substrate. So we end up crystallizing with an analog, right? So what you can see down here, if you look at this, when it's not bound in this dark blue, do you see how much it protrudes into the cytosol? So we have lots of area in this G protein coupled receptor that can associate with other proteins. But what happens when we bind? It kind of gets pulled up into the membrane, right? So now you no longer have those sites that are available for association with other proteins. And so that's the big conformational change that's going to start this whole pathway. Okay. So a lot of times what we want to be able to do is to understand how a pathway works and then to manipulate it, right? That's what we do in science. We don't discover anything new. We just try to figure out how to manipulate things, right? So there are two different ways that you can manipulate this system. You can use what's called a receptor agonist. A receptor agonist is going to bind to that same receptor, and it's going to mimic the natural response, right? So it's so close in structure that we're going to have the same response. So this is the same response as natural ligand. NTU, natural ligand. Right? So this is an artificial way for us to activate the receptor, right? Because sometimes you want to be able to activate the receptor. But think about just general treatment of patients. Sometimes things aren't working enough, and sometimes things are working too much and you need to bring it back down, right? So an agonist will activate a receptor, an antagonist will bring it down, right? So remember, if anti is opposite, right? So a receptor antagonist is going to bind to the receptor, yes, but it is going to block the natural ligand from being able to activate the receptor, right? So this is very similar to the analog, right? It has super high affinity, but the thing is, it does not induce a conformational change in the receptor. Because if I induce a conformational change in the receptor, then I'm a receptor agonist. So a receptor antagonist or an agonist will bind to the receptor, but no conformational change, right? That's the difference. So this has conformational change. This is without structural changes, right? So that we can actually take advantage of being able to hijack these processes in the body. So here are some examples, right, of natural substrates for our beta-2 androgenic receptor. We have epinephrine, norepinephrine, and we have dopamine. All three of these will bind to that receptor. So we can actually make receptor agonist. A receptor agonist, this is an example of an inhaled steroid. So like if you have asthma, right, you get an albuterol inhaler, right? What does an albuterol inhaler do? Right, usually your airway is becoming constricted. And so we want to relax. We want to have muscular relaxation, and we want to have blood vessel dilation. Well, that's exactly what epinephrine, norepinephrine, and dopamine can do. So we are going to mimic that activity by giving you an inhaled drug that will go straight to your windpipe and replicate what the body would normally do if it had epinephrine, norepinephrine, or dopamine. We can do the same thing. We can make something that's an antagonist, something that is going to bind and prevent vasodilation from happening, right? Okay. So, we've talked in this class about what happens when you don't eat and what happens when you activate your fight or flight response, right? So when you don't eat, you make glucagon to get blood sugar into the bloodstream so that you can feed your brain. Same thing happens when your fight or flight becomes activated, right? Because if you're trying to run from a bear that came in from the back door, you're going to need a little extra boost of sugar to be able to run, to be able to see the bear, to be able to breathe faster, to make your heart go faster, right? You're going to need a little influx of sugar. So what we're going to talk about is how Gpcr results in that release of glucose. So that's our start and our stop. So we have two different ligands that can activate the same downstream effect, glucagon and epinephrine. They bind to different receptors. So glucagon binds to the glucagon receptor. Epinephrine, on the other hand, can bind to two different receptors. It can bind to the beta-2 androgenic receptor, and it can bind to the alpha-1 androgenic receptor. Each of the receptors are going to activate different downstream targets. So the glucagon receptor activates adenylate cyclase. Same thing with beta-2 androgenic receptor. So if you activate the same enzyme, you're going to create the same second messenger, right? So we typically function through a similar G protein coupled set of proteins. And we call those G, this is the G protein, GS alpha for stimulatory. The S stands for stimulatory. I'm going to show you these in just a second. But we said epinephrine can function in a different pathway as well. So if epinephrine activates the alpha-1 androgenic receptor, then we're going to activate a different enzyme. In this case, it's phospholipase C, PLC, right? So start to get familiar with those abbreviations. This is going to create what 2-second messengers that we talked about today? DAG and IP3, right? So this functions through a different set of G proteins. So let's go look at those G proteins. Okay. So when you have an enzyme, I'm gonna skip and I'm gonna come back to this. Okay. This is a receptor. This is a G protein coupled receptor. This G protein coupled receptor associates with a set of G proteins. That's why they call it a G protein coupled receptor, because it's coupled to the G proteins. So if you look, there are different subunits to the G protein. We have an alpha subunit, we have a gamma subunit, we have a beta subunit. The alpha subunit is typically bound to Gdp. But when the receptor binds and it activates that receptor, what's going to happen is that this alpha subunit is going to exchange Gdp for Gtp. When that exchange happens, you disassociate the other subunits from the G protein. And when those subunits disassociate, now we're going to induce some downstream signaling events from occurring. Okay, so let's go back one slide, right? We have this G protein, alpha, beta, and gamma. They're disassociated because they're active. This alpha subunit has Gtp. So there are different alpha subunits, and the different alpha subunits are going to do different downstream effects, right? So what did we say this was going to activate? GS alpha, right? So let's go to GS alpha. Here's GS alpha. What is GS alpha going to do? It's going to activate adenylate cyclase, right? So that's exactly what we said. It's going to activate adenylate cyclase. It's going to make cyclic AMP. And remember that pathway that we talked about making tons of glucose from glycogen? That's exactly what's going to happen. Okay. So that's adenylate cyclase. But do you see how there are many different alpha subunits? And each of the different alpha subunits has a different effect. So if we go back and we look, what do we have for our alpha-1 androgenic receptor? GQ alpha, right? So let's go to GQ alpha. What does GQ alpha do? Stimulates phospholipases. What is a phospholipase? Why would that be important? It's an ACE, so it's an enzyme that's going to cleave phospholipids. When I cleave the phospholipid that's embedded in the membrane, what second messenger am I going to get? DAG and IP3. Okay, so depending on which alpha subunit is bound to the receptor will tell you what downstream effect you're going to have, right? Because there are lots of different G protein coupled receptors. Let me skip ahead. See how you have different senses, right? We said all of the senses are controlled by G protein coupled receptors. So if they're all G protein coupled receptors, why don't they all have the same effect? Well, it's because you have different subunits associated with it. If you're talking about vision, it's the rhodopsin G protein coupled receptor. And so this has a GT alpha subunit. The taste receptor has a GQ alpha. The olfactory has a GS alpha. So do you see how they're different? Yes.
Is there a difference in the receptor itself at all?
Yes, because they have to respond to different stimuli. Yeah.
They just fall in the same category. They don't have the same, let's say, coding.
Correct. You're going to have some similarities because you are going to have that serpentine, but what's going to be really, really different are the cytoplasmic side and the extracellular side. So the extracellular side is going to have to be unique to the ligand, and then the cytosolic side is going to have to be specific interactions with the specific G protein coupled G proteins. Yeah, exactly. Okay. So what happens in a general mechanism for G protein coupled receptor activation? You have the ligand bond. The ligand induces conformational changes in our Gpcr. When that happens, you have a receptor mediated stimulation of what's called a guanine nucleotide exchange. Why is it guanine? Because it's guanine. It's Gtp being replacing Gdp. So if you look over here, you see how we had Gdp attached? When this receptor is activated, Gdp is swapped out for Gtp. So this is a guanine exchange factor that will facilitate that exchange. Then you have downstream effectors, right? And they're regulated by whatever complex it is it's associated with, right? And then eventually you're going to have termination of the signal because you're going to put that GDP back. If you never put the GDP back, then that receptor's on forever, right? And that can be a problem. That's a really funny story. You want a side story? So you know my birthmark, right? So this is a mutation in a Gpcr receptor. So exactly what happens is because of this mutation, the cells continue to get the signal to make blood vessels, right? And so it never exchanges back to GDP in these cells, which just continues to make blood vessels. So about every year to every 2 years, I go and have laser treatments on it. I had a bunch as a kid, a bunch. But now just to kind of keep it in check because it continues to grow. So it can be a real problem, right? It's a really interesting thing because in development, it's a spontaneous mutation. Like they found this out when I was in graduate school and I was finally able to tell my mom, look, it's not your fault, right? It's a spontaneous mutation, you know, it's just a mistake. And so depending on when in development you get the mutation, right? If it's on the head, right? The same cells that develop into skin cells also develop into brain cells. So depending on when in development the mutation occurs, will tell you if you'll have neurological defects or not. So when I was born, all the doctors told my parents, she is going to be mentally handicapped, you know, you need to prepare yourself for this, all this kind of stuff. And I love my mom because she told the doctor, there's the door, don't let it hit you in the ass on the way out. And that's just who she is. And so they didn't know that then, right? That was, I'm not going to say how many years ago. That was a while ago, right? So basically, so if you look here, right, you have to have an enzyme that is going to exchange this Gtp back to Gdp. So you have one enzyme that goes from GDP to Gtp, and then one that goes from Gtp back to GDP. Yes.
How exactly does your laser treatment inhibit?
It doesn't. It doesn't. It kills the blood vessels. So it's just, it goes in and it fries them. It's super painful. So it's what you would have to have if you have a tattoo and you decide that you don't like your tattoo anymore. Let me tell you how much it hurts. Think twice before you get a tattoo, especially on a very sensitive part of your body. Because trust me, getting a laser treatment on your face sucks. All right. Any other questions? It was a good side trip. Don't get tattoos. So everybody's like, do you have any tattoos? I'm like, no, because I know exactly what it feels like to get rid of them. No. Exactly, exactly. All right, so let's get back to our pathway, right? What was our downstream signaling protein? So go back and look at, I know I keep skipping around, right? But look at your enzymes that are being activated. So if we have adenylate cyclase, we have phospholipase C, what's going to happen? What are those going to activate? So let's see, I have to go like all the way back. All the way back. Good Lord, here we go. Right? We're activating PLC. PLC is going to activate DAG. DAG is going to activate protein kinase C. Protein kinase C phosphorylates downstream target protein. So this is kind of where we are in the pathway, right? So where was I? Oh my goodness. Okay, so we got PKA. PKA becomes active when cyclic AMP levels are high. So when you think about this, right, what do we want to happen? If we are in a starvation state, we are in a fight or flight state, what do we want? We want glucose in the bloodstream, right? So there are a couple of different ways that we're going to get to that point. We're going to do a few things to make that one thing happen. So the first thing is we want to inhibit the body from making glycogen, right? Because if we want to break glycogen down to glucose to get it out into the bloodstream, we want to stop making it, right? So you have to inhibit the glycogen synthase enzyme. So you stop making glycogen. Then you're going to also activate enzymes involved in glycogen degradation. So we're going to break glycogen down and stop glycogen from being made. And at the same time, we're also going to activate enzymes in a separate pathway that will get us glucose. The other way that we can get glucose is through gluconeogenesis. So you actually create glucose, right? So all three of these things are going to result in what? Increased blood sugar. And that's the whole point. That's the whole point of releasing epinephrine and releasing glucagon, is so that we can increase our blood sugar. Question? No, ask. It's great.
Can you explain what the first, like out of the three, what was the first one?
Oh, phosphorylation. So okay, so this is a kinase, right? So its activity is always going to be phosphorylation, but it has actually three targets. The first target is glycogen synthase. Glycogen synthase makes glycogen, right? That's our storage molecule for glucose. So I don't want to invest glucose into making glycogen, because then that pulls glucose out of the bloodstream. So I want to inhibit that process.
It says phosphorylation, so does the phosphorylation inhibit?
Yes, yes, the phosphorylation inhibits glycogen synthase. So you are going to inhibit the making of glycogen.
I guess when you usually think of phosphorylation, you usually think of some type of activation process.
But it's not always.
Oh,
It's an on-off switch, right? You know, like if you have two different light switches in your house, right? Sometimes up doesn't always mean on.
yeah, like if you have two of them that are the same circuit on opposite sides of the room.
Correct. It's the same thing. So sometimes phosphorylation is turning it on, sometimes phosphorylation is turning it off. Just depends on the protein. Yep. So we're going to phosphorylate glycogen synthesis. We're going to phosphorylate, and we're going to activate enzymes that will break down glycogen. So this will make glucose. We'll release it out into the bloodstream. And then we're also going to make glucose via gluconeogenesis. So three different things are going to happen, and all result in an increase in blood sugar. This doesn't actually increase blood sugar, the first one. What does the first one do? It prevents a decrease in blood sugar.
Because you don't want to be hauling water out of a boat with a hole in it.
Right. Exactly. Okay. So now let's look at PKA's role in epinephrine, right? This is where we bound to the beta-2 androgenic receptor. Remember that? So we have cyclic AMP, right? This is our second messenger. Once our second messenger is activated, it's going to go in and it's going to activate pKa. What does pKa do? We said three things. It turns off glycogen synthesis, it turns on glycogen degradation, and it turns on glucose synthesis from scratch, which is gluconeogenesis. And all of those end up in you shipping glucose out to the bloodstream. All right? So this is just a visual representation of this. Yes, ma'am.
So for this, the ligand is
Glucagon. Well, so this is the beta-2 androgenic receptor. So go back to your table, your beta-2 androgenic receptor. So the beta-2 androgenic receptor is going to be activated by epinephrine. But glucagon, when bound to the glucagon receptor, will activate the same enzyme, the same second messenger, the same downstream targets. So technically, because it's beta-2, it's activated by epinephrine, but it has the same response as glucagon binding to the glucagon receptor. Yep. Yep, you got to kind of be careful about those things. All right, where were we? All right, so now here's our alpha-1, right? This is epinephrine binding to our alpha-1 receptor. So how is this different? If you go back and compare, right, we have a different set of upstream signaling occurring when we're talking about a beta-2 versus an alpha-1. Our upstream is going to be different. So in upstream signaling for this one, we're going to activate PLC. PLC is going to hydrolyze those phospholipids that are stuck inside the membrane. And we're going to make two second messengers, diacylglycerol and IP3. Because we have these two second messengers, now we can go on and we can activate PKC. And PKC is going to go in and turn off glycogen synthesis. The IP3 is also going to mediate that calcium release that we saw earlier, which eventually also leads to glycogen degradation. So we're going to get the same effect of glucose being shipped out to the bloodstream. So we're going to end up having a high blood sugar, right? And that's what we want. So we're saying that no matter what receptor epinephrine binds to, we're going to have an increase in blood sugar. But it's going to follow a different set of pathways. Why is it good to have this kind of redundancy? It is redundant, right? What is your most important organ in your entire body? I would say brain. I would say brain. You can get a replacement heart. You can't get a replacement brain. That's my qualification there. So your brain's primary food source is glucose. The rest of your body can feed off of other food sources, but your brain primarily uses glucose. It's not only one, but primarily is glucose. So if you start starving your brain, you are going to have serious problems, right? You are going to go into a coma. You are going to die. So redundancy is a good thing. That's what I'm trying to say. Okay. So how do we control this signaling cascade? We control it with GEFs and GAPs. Of course we do, right? We have to have some more abbreviations because you didn't have nearly enough yet today, right? So a GEF is a guanine nucleotide exchange factor. A guanine nucleotide exchange factor promotes the exchange from Gdp to Gtp. So what does that do to signaling? Does it shut it down or does it activate it? It activates it. It activates signaling because now these things disassociate. So do you see here, we have our dissociation. Dissociation leads to signaling, right? And depending on which subunits you have, will tell you what kind of signaling you have. Okay, and that's fine. They're disassociated. If they stay that way, you're going to continue to signal. It's never going to stop. But what we need is a gap. A gap is a GTPase activating protein. What that does is it activates the GTPase activity of our G protein. So we have GTP bound to our G protein, and we have this gap. This is the gap. The gap is going to tell this G protein, hey, dude, hydrolyze that GTP into, hydrolyze that GTP into GDP. So once we hydrolyze it, now everything reassociates and we go back to the resting state.
So when the Gpcr is activated, the old GTP molecule is kicked out, a GTP is put in, and then we turn it off, and that GTP is turned into a GTP.
Correct. It's hydrolyzed. Yes. Yes. So my mutation is actually in a gap. Isn't that cool? All right, y'all have a great weekend. And start studying G protein. It's a bit confusing. Gaps and gaps and all the G things.
Quick question.
Sure.
I promise it's
No. Oh no, and I didn't put on my chat today. Y'all gotta help me with that. Okay, anybody who needs anything on Zoom? I was not monitoring the chat. Oh, I'm so sorry. Anybody still on? Yeah. Okay, but it doesn't look like anybody needs anything. All right, I'm gonna go ahead and end the Zoom.